Hi, everyone, and welcome. My name is Caitlin, and I'm the Communications Manager for the Pediatric Cancer Data Commons, or PCDC. Thank you all for joining us today, and we're very grateful to Dr. Resnick for joining us for this third installment of our guest speaker webinar series, The Common Good. The purpose of this series is to hear from thought leaders across the fields of pediatric oncology, data sharing, and international collaboration about big ideas and opportunities for change. Just a few housekeeping notes before we begin. This webinar is being recorded, and we will send out the link to that recording to everyone who registered later this week. And all the recordings from this series are gathered on our website and our YouTube channel. So if you missed the earlier talks in this series, you can check them out as well. We have videos of our talk from Greg Simon, the former president of the Biden Cancer Initiative, who talked to us about clinical trials, and from Sam Blackman, the co-founder of Day One Biopharmaceuticals, who talked about how drugs for pediatric cancer treatment are developed. And I'll include a link to that archive in the follow-up email. If you have any questions for Dr. Resnick, we'll have Q&A at the end of the talk, so you can use the Q&A function in the bottom bar of your Zoom screen to enter questions at any time during the talk, and then they'll be addressed at the end. And with that, I'll hand it over to the director of the PCDC, Sam Bolchenbaum.
Thanks, Caitlin. It's really, really exciting to be here today and to be able to introduce Adam. I've known Adam for a long time. He's the director at Children's Hospital of Philadelphia of their data-driven discovery and biomedicine group. And what's cool about this group is that it really is across the entire spectrum from biospecimens and clinical research and molecular diagnostics, preclinical work. And so Adam thinks about these kinds of problems in a very large way. And so he's thought about his work focused on brain tumors. But the cool thing about Adam is that he takes this way beyond CHOP now, and he works with several brain tumor consortia like the Brain Tumor Tissue Consortium and PNOC, the Pacific Pediatric Neuro-Onc Consortium, and is thinking about better ways to develop clinical trials for kids with brain tumors. I know Adam through his work with the Gabriella Miller Kids First Data Resource Center, where he's truly a visionary in thinking about how we can think about pediatric cancer data in terms of a developmental disease. And so the Kids First DRC is collecting not only pediatric cancer data, but also data on developmental disorders, and being able to combine those two together is truly a new way to be thinking about cancer research. With the Seven Bridges group, he co-founded Cavatica, which is an amazing analytics platform for looking at genomics data. It's just been so fun knowing Adam over the years and watching the way he thinks and the way he looks at these large problems. And so I'm super excited today to hear him talk about the infrastructure to advance research for kids with brain tumors. Obviously, as I'm sure Adam will talk about, brain tumors remain a largely unsolved problem in pediatric cancer, and remains a huge issue. And so to hear Adam put his visionary spin on this work, I think is super exciting. So with that, I'll turn it over to Adam and let him get started, and looking forward to the talk and hearing the discussion afterwards. So thanks very much, Adam. It's all yours.
Thanks so much, Sam, for the invitation. It's really an honor to be here. Likewise, I'm a huge fan of the work that the PCDC has been doing. And as you mentioned, I have the privilege of both, being scientific director at the CBTN and of PNOC, two organizations who are committed to partnering with PCDC. And likewise, you've been, really instrumental in the work we've been doing across Kids First. And this is really an exciting time. I'm so glad that you're putting together this series, in part because this is a rapidly evolving framework for the pediatric landscape in ways that, although not fully appreciated, the pediatric community is really innovating around how to do science, leveraging new technologies and collaborative frameworks that are both existentially required, but actually required more broadly beyond pediatrics to drive new ways of doing discovery. So as I mentioned, I'll try and take sort of a framing that spans both pediatric brain tumor centered view, but really within the context of what we think is really required to drive pediatric cancer discovery and clinical translation more broadly, and try to contextualize this within some of the emerging frameworks that are both Consortia enabled as well as NIH enabled, and maybe even do a couple of sort of live demos of some of the platforms for folks who are on the line. So my own trajectory is really, I was trained in neuroscience and really began my career with an interest in leveraging that developmental neuroscience framework on behalf of making discoveries in pediatric brain tumors. And at the time when I started my career in 2007, and still now, it turns out to be the case that pediatric brain tumors are the leading cause of disease-related death in children. This is, the most recent registry-based data, highlighting this across both the 0 to 14 age group, as well as all the way up to 39, the AYA age group. And this is something that's, not ubiquitously understood or recognized, and in part because pediatric brain tumors still are considered a rare disease, despite having this framing of impact on pediatric childhood health. But the other part that's challenging about brain tumors is that it's a little bit of a misnomer to say brain tumors. When you say brain tumors, you're likely talking about more than 100 different types of diseases. And so when you start actually breaking that out even further, the challenges around the coordinated discovery process that leverages new technologies and new modalities of discovery becomes increasingly challenging, for any young investigator who sort of naively, moves to Philadelphia to try and start a lab on pediatric brain tumors like myself, back then or now. And that has to do with accessibility to the required resources to drive new discovery, beginning with samples, infrastructure, and data. From very early on, each time we encountered a challenge, and I'm ubiquitously going to use the term we because by and large, all of this work is a collaborative venture. That challenge also presented an opportunity to sort of rethink the way science is done, rethink the framing of how to drive new discoveries in ways that are both social, cultural, political, and institutional. And so from the very beginning, when investigators are trying to assess how to drive discovery by leveraging new technologies that harness large-scale data generation, molecular data generation, for example, from biosamples, immediately you come up to challenges of this age-old question still haunting society at large today on the political side of centralized versus decentralized governance of resources. The academic environment, by and large, has adopted centralized views, meaning each institution has its own specimens, its own investigators, its own funding, in ways that really challenges the capacity of any single institution to drive impact on behalf of a very diverse disease landscape in pediatric brain tumors. And so historically, the pediatric cancer research landscape has been a collaborative framework. But collaboration will only go so far if not paired with new technologies and frameworks for that interaction in ways that drives it forward. And in reality, the real question is not about centralized versus decentralized. The real question in the modern age is about connectivity. The real value for data sets does not require centralization versus decentralization. Those are really surrogates for connectivity of data, because what any scientist learns very early on is more data is typically better. And the reason it's better is because each data point, when connected to another data point, adds value to both data points, and by and large drives the discovery process of hypothesis generation and interpretation of data. So it's really about connectivity of data. And once we began thinking about connectivity of data as a framework, we began really adjusting the model for how we thought about building infrastructure for pediatric brain tumors. we recognized that connectivity in the digital framework is non-local. And what's really the challenge is the localization of the analog of biospecimens and its tracking and processing. And so early on, we reached out to partnered institutions to assess whether there'd be willingness to support a centralized biorepository. However, a centralized biorepository that was connected equally to all institutions, that would not necessarily be centralized or owned by any one institution, regardless of its location, but that connectivity could be empowered in ways that engender trust and transparency around the use of specimens. And so we began with 4 institutions in 2011, and today, spanning both the Children's Brain Tumor Network and the Pacific Neuro-Oncology Consortia, and it's over 30 institutions that have partnered with a shared vision in mind of creating centralized resources that are decentralized in access, governance, and use, that optimizes on limited use specimens, but maximizes the reuse of reusable resources. And the framework really fits into a sort of reconceptualization of the scientific method that says we can start with highly curated, annotated, standardized sample collection across institutions. Our job is to, as rapidly as possible as we can, convert those samples into rich data that can be then structured and queried as information to drive new knowledge creation with meaningful use in ways that can be applied to impact. This can be done only through the intersection of these resources across institutions, as supported by an emerging technological framework of platform and non-local resources that intermediate these interactions and coordinate these actions across institutions asynchronously and in a global fashion. Some of the key sort of tenets of such an ecosystem, and I'll start with the theoretical before going into the practical and do a couple of demos, is that in order to essentially meet the mandate of accelerated discovery that harnesses the full potential of data, we came to and modified a few points around principles of data use. One, a data ecosystem must support discovery knowledge creation as a non-local, information-driven, and collaborative global process. So we have to essentially undermine the traditional local framing of data as a resource. Secondly, the ecosystem, must support and maximize the number of users of the data. So essentially, there is a direct correlation between the value you can extract from data and the number of users that can actually access it. And so the system has to optimize if you are to meet the mandate of patients and families and foundations who essentially contribute to the development of these resources. Then your job as a steward should be to constantly maximize the number of users and improve their secure access to data and information, as well as the capacity to themselves participate in the data generation process. Now, those users, in order to maximize the opportunity landscapes, have to be diverse, right? So diversity breeds innovation and discovery. And that means that you have to take into consideration the platforms that you're building in ways that can support very different domain experts and their use of data in different points of the data lifecycle. And as you're thinking about creating ecosystem and platforms around these efforts, you have to support the connectivity and community building among such users. And this is something that's not at all foreign to the PCDC. Really, it's a combination of non-local platforms plus communities plus resources that engenders new opportunities and new modes of discovery on behalf of the pediatric landscape. And at every step, as you're building these resources up, your job as a steward of those resources is to prioritize frictionless flow of data and information. And typically, although we think of data as streaming and frictionless when we sort of use Netflix or watch movies online, in reality, the flow of data as governed and controlled by institutions or other resources oftentimes is all but frictionless. When you start thinking about intellectual property laws and value and propositions that are completely antagonistic to the consent-based practices that we commit to on behalf of patients. And so we began with something like a couple of hundred cases in the first year when we began creating these consortia-based efforts around collection of biosamples and enrolling subjects. And today we're over 4,200 enrolled participants. And just this past year in fiscal 22, even in the context of COVID, we enrolled 348. Paired with the enrolled participants are our family participants. So this includes parents and at times siblings who themselves provide biosamples. Each one of these individuals provides a variety of different biosample types, including germline source material, as well as a tumor material that's collected from the operating room in a standardized fashion. So we'll come back to standardization in a bit. And then these are essentially aliquots that are available for use, not just by the consortia, but by anybody in the world. So you do not have to be part of a consortia, again, if we're trying to maximize users. You don't have to be part of the consortia in order to utilize the biospecimens. So there are more than 51,000 aliquots of biospecimens available. Recognizing the dearth of preclinical models derived from surgical specimens in pediatric brain tumors, we undertook the creation of new models. And again, these are available in real time for investigators for use and reuse. And very early on, as we began distributing samples for project-specific efforts, meaning investigators submitting projects for requesting of samples with hypothesis-driven queries, we also set the mandate to create very deep and as molecularly rich data sets for these samples that could drive new hypothesis generation and new interpretation, with the key that such data generation should be released in as real time as possible in ways that is unembargoed. And additionally, those biospecimens are paired with clinical and phenotypic data that's attached to those biospecimens. And this, although oftentimes, underappreciated because of the technologically affiliated frameworks of biospecimen-derived, big omic data, clinical phenotypic data is probably some of the most valuable paired data sets, in these cohorts, because we're following patients across time, across their patient journey. And so across this time, as samples and data grew, we've had more than 261 projects, only 30 just last year, and more than 150 publication, and are continuing to grow with new applicant members joining the consortia on behalf of these efforts. And essentially, by joining the consortia, you adopt a set of principles, a framing of legal tenets that describe how data are shared and in what fashion and what timelines, and what governance. And then you take on ownership of essentially stewarding the optimal use of these resources. CBTN really exists, as a pediatric brain tumor entity within, a very diverse landscape of other efforts, both NIH-funded efforts with specific data modalities and data generation initiative, as well as disease-specific initiatives. And this is one of the key framings for us from very early on. While at the same time, communities have a need for identity and dedicated purpose. And here, for example, I'm speaking about a specific disease community. So you can see, for example, the Chordoma Foundation or NF2 Biosolutions. These are community who essentially are centralizing around a single disease within the landscape of pediatric brain tumors. What we found very early on is that it's very challenging as a single entity to build the requisite infrastructure on behalf of your specific disease. And so the CBTN from very early on sort of sat and straddled at this tension between creating generalizable resources that could be used by all disease settings, while at the same time providing a foundational framework from which a launch pad could be advanced on behalf of a disease-specific initiative by a community and an effort. And this central challenge between sort of the bespoke and refined and the general, but scalable and robust, is a constant tension of the framing of the work in ways that we continuously try to advance through the development of portals and resources that ultimately led to the development of the Gabriella Miller Kids First data resource. So as you move from sample collection and begin to generate data, which many of us in the field began sort of harnessing, you have to think about platforms. And we're very privileged in 2018 to have the opportunity to take all that we learned as part of working within CBTN and PNOC, launching Cabatica, the Cancer Moonshot, and apply it on behalf of a new initiative that will launch thanks to a little girl with a pediatric brain tumor, a diffuse intrinsic pontine glioma, as it was known at the time, who 2 weeks before her death filmed a video and in frustration really mandated very eloquently for mankind to stop talking and start doing more on behalf of pediatric brain tumors. And as a result, a bill was passed with allocated funding for 10 years to support the creation of new infrastructure that drives that data generation, but information representation in ways that is scalable and goes beyond brain tumors. And as Sam mentioned, pediatric cancer is unique because it occurs in a setting of development. Pediatric cancer is a developmental disease, but again, this constant refrain of the opportunities lost due to siloed initiatives, where there are opportunities to leverage discovery across developmental biology, where self-fate decisions and disturbance of development that might occur in one disease might actually be shared in cancer, and we can actually leverage those, have not been ubiquitously embraced in ways that the academic enterprise and healthcare oftentimes bifurcate. And so the mandate for Kids First was to really advance this idea that development can be a connector for discovery by supporting large-scale whole genome and RNA-seq data generation for pediatric cancer, paired with whole genome and molecular characterization of structural birth defects and diseases of development. And this is where we immediately came upon the need to expand, our portfolio of resources, even as we developed our consortia-based initiatives and began collecting clinical data and generating data sets. Those activities were largely sort of ad hoc coordinated in fashion as we began generating data and analyzed them. But when you're trying to actually sponsor discovery in ways that an uninitiated investigator comes to a portal and tries to ask the question, do you have data on my specific disease or my specific phenotype or my specific data modality, and trying to guess what kind of researcher they are, you come upon a challenge that is both, again, a challenge and an opportunity, right? Because the data landscape of data use spans from those data scientists who sit in front of a command line screen and type code and build pipelines and analytic frameworks that are downstream used by investigators who really want to analyze the derived results from those analyses in sort of a point and click fashion in portals. Those two communities are oftentimes not connected to each other. They're oftentimes very different communities between the clinical or translational framework and the bioinformatics and bioengineering and data science framework. Oftentimes they're not even housed in the same building or place within institutions between wet lab space and dry lab environments. And so early on, as we began building out Kids First, we started thinking about how to potentially not only support this diverse community, but also advance a mechanism for them to connect across these resources. So let me switch just to, for those of you who haven't done it, just to give a brief walkthrough through the Kids First data resource. And this is a platform that's really a collaborative effort with the NIH itself and the Common Fund to really innovate around portals and their development. Thinking of portals for data sets not as places where people go to download their data sets and go work on them separately, but as places where people go to actually potentially drive discovery, use NIH resources to do that analysis in ways that can be non-local and partnered with other investigators. So anybody can log into the portal and explore it. And when you log in, again, you essentially have to choose one type of a standard login. But this login is really to create a saved state for you as a user. One of the really innovative things that the NIH worked very hard on was to establish clinical and phenotypic data as open access data, meaning as long as clinical data was de-identified as required by HIPAA, that data should be made available for open use by investigators, particularly to define cohorts and ask questions about data sets before they have to engage what are called controlled access data. Now, controlled access in the context of Kids First and in the context of the NIH is mediated only on behalf of data security and respect for the consent process. So namely, within this framework, just like as it was in CBTN, any data can be actually accessed by anyone as long as that entity commits to not trying to identify anybody within the study, commits to a set of practices around data use through their institution, and submits a request that describe what they're going to do with the data in ways that ensures that it aligns with patient consent. So distribution and access to data, the only barrier, right, is essentially commitment to a framework of use of data in alignment with consent and patient privacy. So that controlled access data is really focused on the raw genomic data. And the first job that any portal has on its hand is to essentially allow users to create cohorts or essentially bags of their data. And that resource of bags of data begins to span the different types of data sets and uses for it. So let me show you how this happens. So when a user explores a data set within Kids First, he or she can explore the data by simply typing, a certain category of information here, like a study. Oh, I want to study on brain tumors. You can also see that we took upon ourselves to begin standardizing and harmonizing the phenotypic data that was submitted into Kids First by both Mondo and HPO. But almost always, we try to include source data as well. This was a commitment we made very early on to both provide source material as submitted to the Kids First data resource, as well as harmonized data and process data that empower discoverability and connectivity of data. And when you start doing that, you can start actually seeing a couple of things. So one, you'll see, as you, I think I put two filters in here. There we go. So you can choose a study and apply it, and you see here, these are all the CBTN cohorts that I mentioned at the start, and they span large portfolio disease. And you can see that right now, only about 900 or so of the more than 5,000 participants have data, although this is about to change in ways I'll talk about. But you can also essentially explore and combine data, across different types of features. So let's say I'm actually a bioinformatician interested in RNA-seq, so I can also combine queries in really fruitful ways, but I can also save my queries and or share these queries. So once you save it, you can actually also share it with anybody you'd like. I can send it to Sam and they can review it and they can further iterate on it. So from the very beginning, the act of leveraging domain expertise that spans, biospecimen, clinical, demographic, and molecular features can be a collaborative process. Once you define cohorts of interest, the next thing users want to know is, how do I get access to data? So let me show you just with another cohort in neuroblastoma. So when we began addressing neuroblastoma, here you can see actually 2 studies. One is a Kids First study that was part of the Kids First program. But very early on, we began harmonizing data with other existing data sets. There was no pediatric brain tumor data sets at the time, but there were other neuroblastoma data sets in ways that you've likely already heard about from PCDC and work that Sam has done across Target and COG. And so we actually began indexing other data sets as well that don't exist within the portal itself, but exist elsewhere, in which we can communicate and federate across those resources. So once you essentially identify cohorts, you can then come to the file repository. And within the file repository, you have a choice as a user as to whether or not you want to access the raw sequencing data, the or the aligned reads that we as a data resource center generated, the variant calls or the interpretation of that data as processed by public pipelines, which I'll show you in just a second. And then you have a choice of either downloading that data or, again, to drive collaborative frameworks, work on such data in the context of Cavatica. Now, Cavatica is a cloud-based environment that not only supports the workspace environment for a user to download data, but also provisioning of compute and standardized pipelines, or the development of your own pipelines within that framework. And at the same time as we're providing rapid and hopefully frictionless access to data, we are constantly maintaining governance in its provisioning. So even as a user would try to access data sets, you can see here on the right some actions that I am either permitted or not permitted to perform on the data, depending on whether or not that data is controlled access or open access. And that's only because users can essentially define their identity by connecting to services that authenticate who they are and what kind of dbGaP access permissions they might have to a data sets. And once they do that, you can essentially then leverage and utilize that data on behalf of your dbGaP application. As I mentioned, that controlled access application that many investigators are used to is now actually supported by the platform itself. So the platform here is actually connecting to and talking to NIH dbGaP DAC access approval mechanisms that automatically says, oh, I know Adam is supposed to have access or is not supposed to have access to data sets as of yet. And I, as Adam, I'm authorizing the system to know that information so that when I then come back to those studies and explore them, now the system says, oh, you now have access to all this data because I know who you are and I can verify that analysis. And then I can actually choose to analyze these data in ways that allow the users to actually analyze that data. And so I'm technically copying, or actually technically not copying, I'm technically just pointing to these data, again, mitigating the need to have to move large-scale data sets around. The system essentially points to one single source of data and says, great, I know you have access. I can put it in a project. In that project, I can define partners. I'm not a bioinformatician, but Michele and others are. So I, as a scientist, can work with collaborators within my own workspace environment in ways where data access is provisioned to the files I chose. Excuse me. These types of frameworks that supports essentially authenticated, authorized access to data in as seamless as a way as possible, as paired with clinical phenotypic data, really empower new ways of discovery. And even as we were doing this work, we came upon some challenges. Even when you work with controlled access data sets, Kids First now has almost 50 studies in them. What if a user wants to understand and ask the question, which study has a gene with a variant of interest of mine, but I don't want to have to apply for dbGaP access to 50 different studies? So we negotiated and worked with the NIH and the Office of Standards and Policy to come up with a framework that still essentially maintained privacy for individuals, but empower discoverability. And again, we always feel there's a solution that respects both the patient's need for privacy, but their mandate for discoverability and data sharing, which is actually, ubiquitous across our community. So here I'm doing a query across what is an insanely large number of variants. So these are whole genome sequences. And so even just across 19 studies that are released with 11,000 participants, there are more than 300 million unique variants and more than 61 million occurrences. So we had to develop new technologies to actually allow users to query across these resources, but at the same time, allowed users to assess which studies they might want to explore. So here you can see I queried for a gene. And even though I don't see which specific patients has which specific variants, which would require controlled access data, a user can immediately say, oh, there are 15 studies with this particular variant, one study with this particular variant. Then I can click on this information and see which specific 3 studies actually have that variant. And then I can assess via other characteristics of phenotypes or otherwise, whether I'm interested in pursuing access to those data sets. That level of innovation around accessing open access data, but within a framework of governance, extends into bridging across environments. And so users who essentially interact with these types of resources can also essentially look at the derived results of that information in ways that doesn't require bioinformatic expertise. So here, for example, is a table view of that data, and I'm just going to click on one of these subjects. And you can immediately see a lot of information about this one subject. You can see the number of biospecimens that they have, and also the type of data that's been generated for them. As I mentioned, you can choose to actually port this data into a Kavatica workspace and work on it. But you can also immediately look at the interpretation of at least our take on that data as a data resource. So here's the mutational landscape as interpreted by pipelines available in Kavatica. So let me just show you real quickly. If you go to Kavatica, there are public apps, and within that public apps are all the pipelines and workflows that we essentially implement, as well as many more. There are more than 500. But you can see that all the Kids First ERC pipelines are available for reuse or modification. And it's these pipelines that then generate these results. So if you trust those pipelines or trust the results, you as an investigator can begin asking questions at a biological level of those particular subjects, or even further explore it in a much more combined way or holistic way within the context of all other subjects, within the Pediatric Brain Tumor Atlas, or even more broadly, and ask additional follow-up questions. So here we spanned all the way from sort of phenotypic clinical data queries to workspace environment and analytics, which I won't demonstrate on the bioinformatics side, to the querying and use of analyze results within the context of portals environment, both in the context of variants or mutational data. That framework we find is especially powerful for end users in ways that they not only want to use existing data, but want to bring in their own data sets and work with them. It's within that framework that we are now expanding these types of efforts, at least leveraging the pediatric brain tumor resources that we built to date. So as I mentioned, we've sequenced about 1,000 of the more than 5,000 cases within the CBTN. And thanks to a partnership between the Kids First and the NCI, the Childhood Cancer Data Initiative, we have another 4,500 samples being shipped out this week, actually, for additional tumor normal and parental whole genome sequencing, RNA-seq, as well as methylation through partnership with an intramural team at the NCI. Now, all this data, again, as consistent with our consortia-based practices, will be available without embargo in near real time within the platform, harmonized to all other data sets. And the clinical and phenotypic data are areas where we're going to continue to develop and innovate around, working especially with the PCDC to harmonize and standardize those data. And so here you see really an ecosystem evolving across both platforms, NIH, repositories and resources, and consortia-based initiatives that bring together the various stakeholders, both from a governmental side, institutional side, foundations, and partners who oftentimes have founded, by and large, the underpinning of most of the initial work, together in a new implementation for community-based discovery. And as staunch advocates we are around open access to data, that open access is always within the setting of a framework of authentication authorization. And authentication authorization of use of data, as I've tried to highlight, has two sides to it. One, it supports the traceability and assessment of value that you are creating, because once someone leaves platforms with data sets, it's hard to tell, right, whether or not they failed on their analysis or they needed help or where they needed help. But in platforms, you have communities who can constantly support each other. We as a data resource center can work on problems they might encounter with the data. And at the same time, that authentication authorization provides you the capacity to connect to other users. So trusted connection requires identity. And that identity, as provisioned by resources paired with data sets, is a real key foundational element. So connectedness is really about trust in most of our endeavors to date. Interoperability is about harmonization and standards. And this is something that spans almost every data modality. As large a data, over I think approaching 3 petabytes of genomic data that already exists in Kids First, A's, T's, C's, and G's in genomes is still pretty easy by comparison to harmonize when you compare it to clinical phenotypic data as collected by electronic health records and CRFs and studies. And it's really this next wave of opportunity that we're really excited about, is this linkage between, right, these multimodal omic data sets that span multiple analyses, right, both source data, derived interpreted data across whole genome, methylation, RNA-seq, proteomics, and the clinical and phenotypic longitudinal data. The power of longitudinality as paired with point-in-time data is that transformative moment that we're at today. And that requires this diverse ecosystem of portals, knowledge bases, cloud compute environment, and especially data services that harness new modalities for standardization in ways that we really hope to empower in partnership with the PCDC. And this is in part through regulation. The 21st Century Cures Act now mandates that electronic health record employ standards like FHIR or Fast Healthcare Interoperability Resource to structure and communicate across resources, essentially unlocking the movement of data from the EHR in ways that can make it interoperable. And there's a very large community that's driving the implementation of FHIR and commercial and non-commercial stakeholders supporting it in ways that are driving, again, community-based technology that's paired with community-based standards and agreement on them. And that combination, right, of community and platforms are the new opportunities that we are very excited to drive forward moving onward. And the reason for that is because right now, the process of collecting longitudinal clinical data on more than 6,000 subjects is a very challenging one, right? Each coordinator at each site has to keep track of which patient is enrolled, and at a 6-month interval that they're mandated to essentially refresh their data across all their cohorts of subjects and to support that human-based abstraction into a clinical reporting form. But technology, again, is providing opportunity to advance this in ways that implementation of FHIR and pilots that we're just now beginning to evaluate support the essentially automation and standardization of streams of data, again, as authenticated users to electronic health records, that once you essentially map and code those standards in once, that data can continue to feed and stream perpetually, right? And that perpetual longitudinal data as linked to diversity of multi-omic and other data set in ways that's both human and machine readable, really set to advance into the next stage in ways that we're very excited about. I ended with FIRE because those streams of data essentially presage the real unmet need of patients and families. Accelerated discovery process as part of a scientific effort is always a mandate, but the real challenge for patients with brain tumors or other diseases with no standard of care that's therapeutic is time in ways that the clinical experience and the research experience become interwoven and proximal. And so the real need for most patients is the need to maximize discoverability on behalf of their own care, right? Through data-driven resources, while at the same time contributing and participating in the ongoing development and contribution into the larger ecosystem of data resources. That process is only now really coming to fruition in ways that, thankfully, the NCI is beginning to participate in through an initiative called the Childhood Cancer Data Initiative, the Molecular Characterization Initiative, which, for the first time, really supports standardized clinical sequencing on behalf of initially brain tumors across the nation in ways that standardizes data generation, standardizes workflows for the data integration, but instead of 4,000 samples being sent at intervals of 5 or 6 years for data generation, this data is being generated in real time on behalf of patient use and contribution for research. This is a really novel and innovative framing that will begin to press and challenge current infrastructure in ways we're excited to actually test, vet, and work and partner with whoever on further developing and harnessing this on behalf of the real-time integration of both data generation on behalf of clinical use, but also that longitudinal clinical phenotypic follow-up and the availability of that data for meaningful use in near real time on behalf of patients and researchers. That asynchronous use of data is a real challenge. Data streamed from multiple endpoints coming together in ways that make sense and is trusted is one of the real key challenges. Pairing that with standards that support the real world data use for virtual cohorts of arms that mitigate the need to have control arms or large scale cohorts for rare diseases is another opportunity in ways that we hope the PCDC will further empower. And this really brings to bear the requirement for new models of clinical trials. We cannot have clinical trials that take 15 or 20 years to complete to accrue enough patients. And the only way to subvert that is by having a more robust measurement system that measures the impact of care or variables of a trial in much more real time, with the only output not being survival, but other factors that essentially assess the impact of a therapeutic intervention and the capacity to modify that around much smaller cohort in a distributed fashion, leveraging real world data as much as possible. And so with that, I'll really pause and thank all of you for your attention. We're always really open for feedback and suggestion, critique, opportunities to essentially improve on what we're doing within Kids First or the CBTN. It is still a work in progress and will be some time before we fully optimize it. But the only way to optimize it is really through partnership and collaboration. So thank you for your attention and for all of those who have partnered with us, and especially the patients, families, and foundations who have made almost all of this possible from the very beginning.
Thanks, Adam. That, I've heard you talk about this many times, and I learn every single time when I listen to you speak. People should enter their questions in the Q&A box or in the chat. And while we're waiting for people to get enough guts to ask a question, I have a couple of technical questions, but I actually wanted to ask you something I think deeper than that, which is how have you thought about the participation of parents and families outside of the context that we're used to, which is the hospital-driven clinical trial consent process? Because there's obviously, as you know, this growing technology where we can approach patients and families directly, have them give us access to their data, where we can subvert the whole process at the hospitals. And that has varying degrees of popularity depending on who you talk to. And I'm sure you've thought about it. So what is the opportunity there? Is it something we should be thinking about?
I definitely think so. And I think there's increasingly, we've been getting, we just had a meeting day before yesterday, with a family that essentially said the following, which I think is the ubiquitous framing for this. All of these families essentially have communities on Facebook or otherwise, that they both partner with for support and navigation, but also to share and exchange data, right? They share personal medical records and others and experiences within this framing. And once they begin to understand the value of data and they recognize that maybe a researcher doesn't have access to that data, there isn't a mechanism for that data to come to that researcher, there isn't the required institutional agreement for that process. And once they recognize that per the Office of Civil Rights, they actually have the right to distribute themselves or mandate distribution of their data from any clinical source, they feel empowered. What's lacking still is the technology that fully empowers that on their behalf, but everything is in place to make that possible as a framework in ways that, to your point, and I remember sitting there, as part of an Obama administration review of these sort of legal underpinnings for ownership of data by patients. The lawyer essentially said, we allow people to do much more dangerous things in our society than share data. I think she gave the example, we allow them to jump off of buildings without a parachute and just some flimsy wings. Patients can share their data with their great uncle or their student, whoever they want. It's their data, as long as they do that in a way that's under their control. And our job is to inform them and support them on that behalf. And so I think it's a moment in time where the opportunity for technology is just about there to empower patients themselves as communities to coordinate the distribution access of their data, particularly as more and more richness occurs at the clinical interface around data and biospecimens in ways that historically have been relegated to the research arena.
So, but given that we're trained to be such good citizens of the old-fashioned ways of doing things, do you worry about alienating or doing things outside of the conventional system that might alienate, that might make us, I don't know, among other researchers, seem like we're a little bit too avant-garde?
I think the way that we've de-risked that process is to have patient communities themselves or patient stakeholders part of the process around this. So essentially, even within the CBTN, we have essentially resident stakeholders as a board that sits, informs, and guides our activities. And when you have really such transparent partnership with patient stakeholders and communities, it becomes much more challenging for dogma or practices that exist for reasons that might be anachronistic at this moment in time to remain. And by and large, most all researchers in the pediatric space, right, are doing this because they care about patients and they care about the families. And so once you empower patients' families with the information, knowledge, and voice for them to advocate on behalf of what it is that they think is right, have the conversation that they want to have. It's less about, me or you being cowboys or, out of the norm, and more about conversation between patients and the community that they're partnered with.
Yeah, I agree. That's great. Thanks, Adam. And to that end, Michael Watkins on our team, who's an expert in fire, asks the obvious question, because you alluded to the technology. What are some of the practical roadblocks that we're seeing now with using fire to stream data out of electronic health records?
So, even though there's a mandate for this, by CMS under the 21st Century Cures Act, implementation by EPIC and Cerner is not quite there, at least by our experience, even the CHOP and most institutions that we've seen. And secondarily, it's not a panacea. The way data go into the EHR don't magically become standardized by just virtue of fire, right? And so there are also decisions and opportunities for the way clinicians enter data into the EHR that can make them much more amenable to fireization, or I like to say burning, if you take the fire metaphor too far. And again, those are opportunities that may initially seem like a tall order to a clinician to change the way that they put in data. But what we find is that, it's true, clinicians don't have time, right, to fuss with data entry or things along those lines. But if in the act of putting in data in a certain way, they actually have more immediate return of value to the care that they provide a patient, they'll always do it, right? And that's the other part, right? Right now, we think of FHIR as a one-way street of taking data out of the EHR. But many of these platforms have the opportunity to make it a two-way street, right? As you put in data, you actually get something back, right? In near real time, right? It shouldn't be sort of discontinuous, right? Like as you put information about your subject, can that help you actually make decisions about your patients in ways that right now, most EHRs are not empowered to do. And I think that's the opportunity at hand to drive a cycle of data curation by the stakeholders themselves, because they are going to be basing their decisions based on the data that they put in ways that would only enhance both for them and the rest of the community. And that's a cycle that's currently broken and missing in most environments.
Totally agree. And that's where the carrots and the different sticks come in.
Yep.
We have a lot of questions now, but I'll try to filter through. So Robert Martin asks something that I was wondering too, is that does everybody in PNOC and CBTN just agree to share all their data, or how do you work out those processes and those embargoes and things like that?
So it's interesting, it is an act of social engineering and a little bit of luck. And so we do have a master agreement. And once we got 4 institutions, so this was Chicago, Seattle, Pittsburgh, and CHOP, to sign that master agreement with the Constitution, there's a community-based process that then drives others to want to join, be a part of it. And because everybody has to sign the same agreement, once you have 4, the 5th one says, well, if 4 of them sign, the 5th one will sign. And so there's a mass action effect. It takes time to get that first process to move forward. But once you do get more than a few sort of stakeholders involved and commit to a set of practices, this is where, in contrast almost everywhere else, we sort of leverage the resistance of most systems to want to change, right? So nobody wants to change the master agreement once they've already all signed it and go through the whole rigmarole. So if you get that right, there's a lot of potential.
All right, I think we're coming up on the hour, and we did a lot of questions, and I want to be respectful of people's time. So
sure.
Just so appreciative of you doing this. This has been a wonderful experience for us, and it's as you mentioned many times during the talk, we're trying to find ways to work together and to be able to connect our systems together. And the technology is really catching up to the things that we want to do now finally. And I think it's an extremely exciting time to be working together, to be working with the NCI, and to be part of the Children's Cancer Data Initiative and other things like that. So we hope to engage you again, Adam, and to everyone who joined us today, thank you so much. And we'll be sending out reminders about our next talk in the series. And we look forward to seeing everybody again soon. So thank you, Adam, very much appreciated.
Of course, and if there are any questions that weren't answered, feel free to reach to our teams directly at cbtn.org or kidsfirst.drc. There are support, essentially, questions or any questions you'd like to connect with us. Our teams would be happy to work with you.
Yep, and I'll send you the questions that I have here.
Awesome.
And, great. And thanks to Caitlin and everybody else for organizing, and we'll see you next time. Thanks so much, everybody.